Hey guys, we're going to be going through topic test 2 today. So hopefully you've already had a go and you've given yourself the 40 minutes and 5 minutes or 10 minutes reading time before you did that. So just like I've posted on Edmodo, it's really important that you guys are completing the topic test and then marking it, either using the assistance of this video or the solutions that are posted, and then that you provide me with that mark for how you've gone. It's going to help with tracking your revision and areas that you need to go through, so that would be greatly appreciated. All right, let's go through this. So we've got the 10 multiple choice to start with. Graphs, I love graphs. Now, if you haven't already, please make sure that you've gone through and as a minimum done the last activity for the graphs worksheet. Goes through and it compares rate of reaction and product on different graphs with different variables on the x-axis. It's really important that you understand the difference between reaction, rate of reaction, and the product that's formed. That's where a lot of students get confused. All right, so let's go through this. So using the following information to answer questions 1 and 2. Now, I always like to annotate things before I even read the questions. So again, we've got the rate of reaction. So that's how fast product is being formed. So product formed. So the rate in which that's occurring. It's not the product that's being formed, it's how fast product is being formed. Okay, when you have product, it's here instead of rate of reaction, this line can't come back down to 0. Okay, because unless that product is then being broken down and used by another reaction. All right, but with rate of reaction, it definitely can come down to 0 because the reaction might stop. It might not be producing any more product. So that's a main point for you to really understand. The substrate concentration is here. Always assume that the graphs start with 0 unless they're telling you something else. So therefore we can see that as we're going along, that substrate concentration is being increased. Now you should already start to be thinking about the factors that affect the rate of reaction of an enzyme. Okay, so we know that already from this graph, substrate concentration, I'm just going to abbreviate them on the side, enzyme concentration. Okay, pH and temperature for obvious reasons of optimal temperatures and optimal pHs. The presence and or absence of inhibitors, okay, and cofactors. If you're not quite familiar on what they are, Dachi has a great podcast. I think it's the prawns episode. Go through and listen to that. He explains it really well. Okay, so let's have a look at what's happening in the graph. So the rate of reaction is increasing quite steadily, and then it reaches a point here where the reaction is still continuing, because remember, if the reaction wasn't continuing, it would make its way down to here, which would be 0. So it's still occurring, but it's reached what we call a limiting factor. Okay, so something is keeping it at bay from increasing any further. All right. So, using the above graph in your knowledge, the limiting factor for the graph is the accumulation of product. I don't see how the product being formed. So remember, we've got reactants, which forms products. The accumulation of product is not going to have any effect on the enzyme and the reactants. Okay, unless it's acting as a regulatory inhibitor, but it's not going to be. The temperature. Okay, if the temperature was increasing, the rate of reaction would increase to a certain rate, and then the enzyme would be exposed to its critical temperature, and then the rate of reaction would come down to 0. Okay, so if the temperature was being increased, we would see some sort of graph that was occurring like this. All right, presuming that was temperature there. The enzyme concentration. Okay, we'll come back to that. That might be plausible. So we're increasing the amount of substrate. So go back to Douchi's analogy, and you've got your prawns. So we're increasing the amount of prawns, but we haven't increased the amount of people that are picking up the prawns. So that definitely could be a limiting factor, because there's only a certain amount of prawns that you can pick up per minute as fast as you're going. So that could definitely be one of the factors. a lack of substrate. If you had a lack of substrate, well, we don't because it's concentrated here. Beautiful. So then we go back and it's C. Okay. Awesome. Next question. I'm going to try and get through these as fast as possible because you guys told me a lot that I talk. Okay. At point X. So I just want to go back for a sec. Point X. I'll try and do that in a different color. Point X is here. Okay. So concentration of substrate has been increased and the rate of reaction is steady. It's staying. It's not changing at all. Okay. The rate of reaction is, now I'm going to draw a graph just so you can see. So the graph that we've been given, it's on the other side of the page, is like this. So remember, 0 here, rate of reaction. So remember, the rate of reaction, if it was 0, it would be on this line here. So the reaction would come, and I'll change it, change colour, would come down to here. Alright, so if it's at 0 and A was correct, that's what we'd say, that's supposed to be an A. Alright, has steadily increased. So then if we were to see it increase, it would keep going up like that. So that would be option B if that was what the graph was to look like. Is at its lowest. So remember, this could be, let's just say this is a 5 up here. It's increasing 1, 2, 3. If it was at its lowest, it would be down here. Okay, and it's not. Point X is over here. All right. For D, is at its highest. All right, it hasn't been higher. So therefore that's point X. So that's correct. It's D. So when you do come across graph questions, please make sure that you understand all of the options and visually what they would look like. Okay, just to help with your understanding. All right, question 3. When investigating the action of a human enzyme, the results shown below were obtained. You know, I like to get you to annotate the graph, so let's do that. All right, so you've got your amount of product of reaction. So again, that's not going to decrease. It's not about the rate of reaction, it's about the amount of product that's been increased. You have 3 tests. You've got test 1, test 2, test 3. Before you even start looking at the options, I want you to make some inferences about what you can see. So the amount of product is over here. Test 1 has a smaller amount of product. So therefore, it would probably have had a less amount of substrate. Less substrate, just write that there, to begin with. Okay, that's one of the options. It also could have been exposed to a lower temperature. Okay, excuse me. It also could have probably had a lower enzyme concentration. All right, so remember all those factors that we were talking about with question 1? They're the sorts of things that you need to start be looking at when you're looking at graphs. Test 2 and test 3, they have the same amount of product that's been produced. So you can see that there. So go across with test 1. Significant difference, but for test 2 and test 3, they're exactly the same. The only difference is the time. We can see that in the trend of the line. Okay. So test 3 has happened a lot faster than test 2. Now again, I want you to start thinking about why. So it could have been exposed to high temperatures. It could have been provided with more enzymes, okay? Things like that. It hasn't been given any more or any less substrate because the amount of product is being the same. Because remember, there's a direct correlation between the amount of substrate that you have and the amount of product. All right, it's just the rate in which that they're going to be produced is the only difference that you will see. So the most valid interpretation for this graph is that test 1 would not contain any enzymes. Well, we don't know that for certain. Okay, like we said, it could have been less substrate concentration, it could have been a lower temperature, could have been a lower concentration of enzymes. Okay, so we don't know that for sure. All right, so we'll move on to B. Region X, just change colour here, is up here. Region X indicates that all the active sites are full. All right, what can you tell me about that? So all active sites are full. If that was the case and we'd saturated the enzyme, we would see a graph that would be like this. So if there was still more substrate to be bound with the enzyme, it would still see more product being formed, but just at a slower rate, okay? Because the enzyme has already been at its maximum capacity, it's been saturated. But if we have more substrate, then we're going to be producing more product, okay? So we're not going to see a plateau in the amount of product that's being formed like we do here. So we can get rid of B. Test 2 may have indicated less enzyme than test 3. That sounds like something that we were talking about. So we'll leave that there. We'll go to D and see if that's what we think about that. Test 3 was undertaken undertaken at approximately 20 degrees, while test 2 was taken at approximately 37 degrees. So they're telling us here in this option that test 2 was exposed to higher temperatures than test 3. If that was the case, then we would have formed the amount of product a lot faster. Okay, so that's incorrect. It needed to be around the other way. So we can cross out D. And now remember, option A, test 1 will not contain any enzymes. We're not 100% sure of that. We can't rule that out for sure. So the most correct answer here is C. Beautiful. Now, for question 4 and 5, I'm going to skip along to the diagram that we can see for question 6. So remember how I said that you need to be able to draw this diagram, label it, and talk about the inputs and outputs for photosynthesis. All right, so you should be able to do that now nice and quickly. So X here is obviously the stroma. Z is the stroma. X is the outer membrane. Again, I'm just going to abbreviate. Y is the inner membrane. Oh gosh, I keep stuffing this up. W is the inner membrane. Y is the thylakoid membrane. Okay, and they haven't asked you to label it as such, but the compartments or the sections that make up the thylakoid membrane is the grana or the granum, and inside that is the I've just had a lumen. Thank you. Okay, so you should be able to label that. Now you should also be able to tell me what stage is what. So here we've got the light dependent. Okay, so remember we had the membrane in our class activity and it looked something like this. So that's the chloroplast, and then we had the protein channels as well. Okay, so that's the light dependent because the light is being absorbed here and by the chlorophyll. Yep. Then remember it moves out, use purple via that ATPase, and then that's moving into the stroma. So therefore that's then the light independent. Okay. The other thing that you should be, I'll just move that here. The other thing that you should be making sure that you write down whenever you see a question about photosynthesis is the equations. So 12 waters, 6 carbon dioxides, we require chloroplast, chlorophyll, sunlight, and then that gives us glucose plus 6 new waters plus 6 oxygens. All right. And you also need to be able to look at this equation. Sorry, it's not the neatest equation, but you need to look at that and be able to tell me the inputs and outputs for the light dependent and light independent. Okay? So you should be doing that by yourself now. So, the site of glycolysis. Oh, I've just labelled a chloroplast and it's not even relevant to it. Oh well. Question 4. Sorry, site of glycolysis. So remember, this is talking about, excuse me, cellular respiration. There are 3 stages of cellular respiration. The first one we have here is glycolysis. Second one that we have is Krebs. And then we have the electron transport chain. Okay? You should know already that these 2 sections here occur within the mitochondria, and therefore this one occurs in the cytoplasm. Oh. The other thing that you should know is the amount of ATP that's created. So you should have a net 2 net ATP. I'm all over the place today. Remember, another 2 here and 32. So it's 2, 2, 32. All right, so that adds up to 36 ATP. And we are going to be talking about that a little bit shorter in the short answer. Okay. So cytoglycolysis is in the cytoplasm, the matrix, cristae, or the and it's A. Beautiful. Okay. Following reactions. So we have protein synthesis, photosynthesis, glycolysis, or Krebs cycle. Which of the above reactions are catabolic? So we have catabolic reactions, and we have anabolic reactions. Anabolic steroids, remember, from estrogen, so it's building up. Catabolic, C for cutting, so it's breaking down. All right, protein synthesis. So that's synthesis is creating, so that's building up. All right, so I'm just going to color code these to make it a little bit easier. So we'll have blue as catabolic and then purple as anabolic. So protein synthesis, it is building it up because it's creating proteins. Okay, photosynthesis, what are we doing? What's the main input? We're creating glucose, and we're doing that from water and carbon dioxide and the light energy. So again, we're building up. Glycolysis, we are taking glucose, and we're breaking it down into pyruvate, and we are creating ATP. So we're breaking down that glucose, aren't we? Krebs cycle, again, we're taking the pyruvate, we're taking off the carbons, remember, we're oxidating it to create more ATP. All right, so it's breaking it down again. Now I've got rid of all of the letters and we can't see it properly. Oh well. Which of the above reactions are catabolic? So we know that it's the last 2. All right, so the only option for that is this one here. So the answer for that is B. All right, question 6. So use the following diagram to answer questions 6 to 8. So remember, we've already labelled it, we've already got our equation. So the stroma is represented by, obviously we've already labelled it, so it's A. The light-dependent reaction occurs at, I can't even read my own writing. Light-dependent, we've got here, that's occurring at Y. So the answer there is C. Chlorophyll would be found in, remember, chlorophyll is here for light dependent, so again, that would be Y, so C. The abundance of oxygen during photosynthesis occurs from, So abundance of oxygen. So this here. Now I want you to go back and think about the visual diagram that we had. Where does that oxygen come from? So if you have a look at this diagram here that I've drawn, remember we started with a H2O in that box, and then we had those 2 electrons there. The light energy came down and excited those, and then they moved along to the next chlorophyll. And now the chlorophylls are lonely. So what it does is it creates that, or causes that water, to split. So then the protons from that, the electrons from that water move into the chlorophyll, and now we have our H ions, which is causing an electrical chemical gradient with inside the lumen. And now we don't need that water anymore. So that water is now a waste product, Okay, so the abundance of oxygen during photosynthesis came from the water. So that's B. Question 10. The primary product of the light-independent reaction. So over here is, so it's asking you what's a product from that, but what's the primary product? Like, what's the most important thing? So primary product, remember we have so when we move out of the lumen into the stroma, we needed our carbon dioxide, and that bound with the Rubesico, which was blue enzyme, and it looked like that. Bind with him, and then we had this 5-carbon molecule. Okay, that bound with that. Then that was unstable, formed a 6-carbon molecule, and then we needed ATP and NADPH to come along and save the day, which then they turned it into those 3-carbon triphosphate molecules, which then go on to make carbohydrates. Most of the time we see it as glucose. Okay, so primary product of the light independent reaction is hydrogen ions. Well, no, we needed those hydrogen ions in order to make the carbohydrates. So no. Glucose. It is one of the main products, so we'll keep that in mind. Water. No, because some of the oxygen that's used turns into water, but it's not the primary one. And ATP, we use that in order to make the carbohydrates. So the answer here is B. Beautiful. Doing well for time. So let's move along to section B. So enzymes such as catalase, you know it's an enzyme because it ends in ASE, which break down hydrogen peroxide to oxygen and water, catalyze this reaction in cells where they are synthesized. Synthesize again just means to create. Where within a cell such as an enzyme, where within a cell is such enzymes synthesized. So the within section here is telling you that this is an intracellular enzyme, and you already knew that because it was in your sac, that is being created. So where is that being made? And you will say that's in the ribosomes, in the cytoplasm. Please make sure that you're specific to cytoplasm, okay? Because there are two different locations of ribosomes, which are on the rough endoplasmic reticulum or in the cytoplasm. If they're on the rough endoplasmic reticulum, that means that they are being synthesized for external use, for other cells. All right, B. If a cell involved in the synthesis of a steroid hormone, so remember steroid hormone is a lipid, okay, because we've got 2 different types of hormones. We've got our polypeptide hormones, which are proteins, and then we have our steroid hormones, which are made up of lipids. Okay, so in a cell involved in the synthesis of steroid hormones is compared to a cell involved in the synthesis of proteins. Name one organelle expected to be in great abundance. So that's saying to you, where are the steroid hormones made? And you would know that as the smooth endoplasmic reticulum, and I'm just going to abbreviate it here, but remember, don't do that. Okay, awesome. Moving along. Question 2. Many fruits, when damaged or cut, turn brown when exposed to air. Whenever you see the word air, I want you to think of oxygen. Okay? When left for 10 minutes, the cut surface gradually browns due to the action of a group of enzymes called phenol oxidases. These enzymes catalyze the naturally occurring phenols of, for example, banana tissue into brown dark melons, according to the reaction. Okay, so they generally always summarize it for you. Structurally, what type of protein is an enzyme? So when they say structurally, they're referring to what level of protein structure are they? So are they primary? Are they secondary? Are they tertiary? Are they quaternary? Hopefully you're aware that the primary structure is the sequence of amino acids, so the order in which they're put together at the ribosome with peptide bonds to form a polypeptide chain. The secondary is the interaction between the R groups, so whether they're hydrophilic, hydrophobic, whether they're repelled, whether they're attracted to one another. So that'll give you your beta pleated sheets and your alpha helix coils, or and then that gives you the tertiary structure, which is assisted by the chaperone proteins, which comes along, which is actually another protein that acts like an enzyme on that protein to actually form it into its 3D shape, which is its tertiary shape, which gives it its functional shape. In this case, for an enzyme, its active site. Okay, so it asks you to explain. So the question here expects you to say that the structure that it is tertiary. The reason why it's tertiary is because that is its functional shape. In this case, it is the active site, which allows it to bond with the substrate, so the phenols, which then allows it to produce the product, which is the melons. Okay, so I'm just going to write it here really quickly. So you need to say that it's tertiary, because tertiary is the functional site, or functional shape, sorry. I think they like you to also include 3D functional shape, which in this case is the active site, which allows substrate to bind with the active site to produce your product. Always make sure you finish off your sentences and be specific to active site, what the substrate are, and what the product are. Okay. B, extremes of heat and pH can permanently change the structure of enzymes. You should be thinking of the keyword here of denaturing. Oh, look at here. Name this change and explain the change in terms of the enzyme structure. So we've already said that this is when the enzyme has denatured. Okay, you would talk about how the increase in temperature or change in pH affects the bonds between the amino acids, which is the peptide bonds. If those peptide bonds break or become weakened, therefore the protein starts to unwind. I like that word, unwind, and therefore it changes its shape. But we're talking about enzymes, so we need to make sure that we're being specific to the active site. If the active site changes, then we are unable to bind with substrate, and therefore no product is able to be formed. Okay, so make sure you're being specific to that. Awesome. The scientists performed a number of experiments to investigate browning of bananas. The banana solution was made and equal proportions were placed into each of the test tubes. Alright, so now I want you to have a look at the table and I want you to see what the different variables are. So you can see that test tubes 1, 2 and 3 have a pH of 7, whereas test tubes 4, 5 and 6 have a pH of 2. So we're looking at pH, but then with each of those tests you have different temperatures. So you've got 5, 25, and 50 for each of those. Okay, so there's some variables that we're looking at. Some of you might say to me that you're only supposed to have one independent variable. That's correct. So what they've done is they have, within an experiment, that's supposed to be an E, you can have different tests. Okay, so this might be test 1, this might be test 2. Within each of these tests, you're able to have its own independent variable. As long as all the other variables have been kept the same, you're able to then compare that data. All right, so with test tubes 1, 2, and 3, you're keeping the pH the same, but then you're only changing the temperature. So for that test, the independent variable would be the change in temperature. Okay, hopefully you can see that. Let's have a look at the results. So we're looking at the colour of the tube in after 10 minutes. Okay, so remember, we're having a look at the browning of the bananas. So the darker the colour change that's occurred, the better that the enzymes are able to work. All right, so we've got a light brown here. I might colour code this actually. We've got a light brown. We've got a dark brown. We have no change. No change. Light brown. No change. So hopefully you can see that the condition in which was the most optimal for that enzyme was where the darker brown occurred. Okay, so we have a look here. It's test tube 2, pH 7, temperature 25. So that is the optimal range for that enzyme because we saw the most change. Okay, we're still able to see change here at pH 7, temperature 5, and then the same here with the test tube 5, so, pH 2, temperature 25. Okay, so you've got the similarities between the two 25s with the same temperature, and then you also see a colour change within the same pH at a lower temperature because the enzyme hasn't been denatured, it's just been slowed down. Okay, beautiful. Make sure you do take the time to unpack your results, especially in your SACs. If you don't understand what your results are and then you get halfway through your discussion questions and then you finally figure out what you are talking about, it might really change your game plan or your strategy towards it, your attitude. So, using the above information, what requirements are necessary for the optimal rate of browning to occur? Now, you already know that it is the pH of 7 and the temperature of 25. So you just write that down. Okay, beautiful. Now, it's asking for 2 marks, so always talk about your results. So I would say that the optimal range of browning has occurred at pH of 7 and the temperature of 25. We can see this because that has the more evident colour change. Okay, awesome. I think I should leave the results table there. In another experiment, test tube 1 and 2 were kept in an oxygen-free oxygen-free environment from the beginning. So remember, oxygen-free. So when you think of oxygen, what do you need to start thinking about? Cellular respiration. And what does cellular respiration give you? Energy in the form of ATP. So, in another experiment, test tubes 1 and 2 were kept in an oxygen-free environment from the beginning and only nitrogen and carbon dioxide gas were present. What changes would you expect in the results and why? So, I'll just pan up, so results, so test tubes 1 and 2, these ones here. You're being asked, what results would you see? At the moment we're seeing a light brown and a dark brown. But we're not exposing this reaction to any oxygen. So therefore, cellular respiration can't occur, and we won't have any ATP. And you're saying to me, well, why does that even matter? Well, all reactions occur because they need energy. Remember, activation energy. So if we don't have any energy to start the reaction, will the reaction occur? No. Okay, so if we don't have any activation energy, no reaction can occur. If no reaction can occur, what will our results look like? We'll say no change in colour. So we've told them what change would you expect in the results and why. I need to explain that a little bit more. So you need to say that chemical reactions occur because they need activation energy. There's no cellular respiration occurring because there's no oxygen. Therefore, no reaction can occur and we won't see any colour change. All right? Hopefully that makes sense for you. Alright. Question 3. Below is a diagram representing cellular respiration in eukaryotic cells. Tests have been carried out using various inhibitors of cellular respiration. By doing this, the ATP produced from various stages can be assessed. The illustration of cellular respiration below is incompletely labelled. This diagram looks horrifying, but I promise you it's really fun. So, let's go blue. We can see here that they've got the membrane of the mitochondria. We know that there are 3 stages of cellular respiration. We have glycolysis that occurs on the outside of the mitochondria. We have Krebs, and we have electron transport chain. Okay, you know that glycolysis gives you 2 net ATP. Krebs gives you 2 ATP. And electron transport chain 32 ATP. In total, 36 ATP. Okay, so let's have a look at the diagram. Electron transport chain has already been labelled for us, which is here. Oh, there's another membrane that I need to make you aware of. Here. Alright. So, remember, this membrane here that the electron transport chain occurs on is the cristae. And that is the membrane, inner membrane, and so therefore in here is the matrix, and that's where the Krebs cycle occurs. Okay, we know that Krebs cycle has an output of carbon dioxide. Okay, we also know that it has an output of 2 ATP. So one of these labels here must be the Krebs cycle. Just try and... So we know that pyruvate is an input for the Krebs cycle. We know that we produce 2 ATP and then we also carbon dioxide. Okay, awesome. So what else do I need to label before we go through this? Electron transport chain. We know that water is produced as an output, so I'm just going to put that here somewhere. It must be occurring there. And then we know that we've got the 32 ATP. Now I'll just keep this up here and I'm just going to read through the questions so that we can just see the visual. Alright, so complete the diagram by adding the following boxes below. The number of ATP is, so at I, so here, which is up here. Let's see if I can colour code this. So we'll do I in red. So I here, remember, it's 2 net ATP. Because how many did we require in the first place? So over here it's not asking you, but we required 2 ATP which gave us 4 ATP. But because we started with 2, we created 4, we now have 2 net ATP. Okay. Double I, which is here. Okay, so that's for the Krebs cycle. And we know that's 2. 3. Now, how many ATP does electron transport chain give us? 32. And then our total is 36. Now, the answer sheet that I've posted on Edmodo talks about 38. I'm going to talk about that when we come to section B. So don't worry about that too much. I would probably expect that you do write 36. Okay. So like I said, I'm going to keep this here while we talk about the next section. We'll go down and colour code it. So just... Alright, so process V, which is here, we know is glycolysis. Okay, and then process VI, which we have, let me find it, VI is here, which we know is the Krebs cycle. Okay. Alright, let's colour code the next one. Yellow. It's a little difficult to see. No one. The name of the site at VII, which is here, and we've already labelled that as the matrix. Okay, and then V3I, alright, which is the electron transport chain, and we know that that occurs in the crystone membrane. Okay, so I'm just going to write that in. See how when you label a diagram already, when you come to answer the questions, it makes it a lot easier. This is the strategy that I used when I came across exams, or I still come across exam questions as a teacher. It's up to you what you choose to use. Alright, so the inputs and outputs at, let's use this pinky colour. So IX, where are you IX? Here. Okay, so the input output, so that's coming in. Remember, what does the electron transport chain need? It requires oxygen because it's aerobic. Okay, does Krebs cycle directly require oxygen? No. Alright, it must be present, but it doesn't require it directly. So it needs the outputs as inputs from the electron transport chain, and because of that, oxygen must be present for the electron transport chain to occur. Okay, so oxygen is going to be present for electron transport chain to be occurring. Those outputs are inputs for the Krebs cycle. Okay, so oxygen needs to be present for that to be occurring. Alright, the next one is X. Alright, so remember, oxygen comes in, binds with the hydrogen ions, and then gives us water, which we've already labelled there. Okay, I think it's a really nice summary diagram. I like it. Beautiful. Alright, so it's section B that I said I'd talk about. There's a multiple choice question that you might have already come across in your weekly questions that addresses this. Excuse me. So what is the net output of ATP? Explain. So, cellular respiration in animals and plants occurs at the mitochondria. Alright? It actually requires an extra 2 ATP for us to move into the mitochondria. Alright? Whereas bacteria don't require that extra ATP to get into the mitochondria because they don't have organelles, because they're prokaryotes. So you might see that it's written as 38 ATP, but in fact we get 36 net ATP because of those 2 ATP that's required to get into the mitochondria. So the response that they want you there to say is that we have 36 ATP, net ATP. All right. The reason being is because we produce 38 ATP, however, we require 2 ATP to get into the mitochondria. Now, I don't really expect that, or hope that doesn't pop up on the exam. I think it can be a bit of a difficult concept, maybe for the higher end students. I don't think they'll really ask that on the question, but it's good for you to know anyway. Beautiful. All right. Question 4. So, what is the name of a process question 4? So we have an enzyme that's over here. I really wish that these were coloured. Enzyme that's here in purple. Okay. Then we have this molecule I, which you can hopefully see is complementary to the enzyme. Okay, and that complementary section here is the active site. Okay, once molecule 1 binds with the enzyme at part 2, which is the active site, produces product X. Okay, which in this case, unfortunately, is an allergic reaction. So, let's go to the questions. What is the purpose of producing a specific chemical to suppress the functioning of an enzyme? What is the process of producing a specific chemical to suppress the functioning of an enzyme called? Now, this is something we haven't directly come across. It's been squished a lot out of the study design, so I don't expect it to really pop up in the exam, but it's, again, good for you to know. This is called rational drug design. Okay, so rational drug design is when they look at the structure of either the enzyme or the molecule and inhibit its function. Okay, by creating a molecule that's complementary to its site, which we'll go through now. B, what is the name given to molecule I? So we would call that the substrate. And again, with the labeling and unpacking the diagram, we've already done that. Part 2 of the enzyme is obviously the active site. Describe how, through the use of a specific chemical, the production of X, product X, could be prevented. All right, so this is leading back to the rational drug design. A diagram may be used to assist your answer. So, I'm just going to start up here, and I'm going to use a light blue. So, to prevent product X, because remember, we don't want product X because it gives us an allergic reaction. So there are 2 ways in which that you could go about preventing this. You could either create a chemical, so remember a specific chemical that is complementary to the active site. If it's complementary to the active site, then it prevents molecule 1 from binding with that. If, again, so remember the substrate, molecule I, if substrate can't bind to the active site, we can't produce product, which is product X. Okay, so you could either attack it that way and go with complementary molecule for the active site, or you could go with a complementary chemical that binds to molecule I. If molecule I doesn't have this binding site exposed, it's not able to bind with the active site. If it's not able to bind with the active site, no product is being produced, in this case product X. Okay, sounded a lot harder than what it is. Really interesting, but it's not that difficult. All right. Last question. So, electron carriers are important in both stages of photosynthesis and all the 3 stages of cellular respiration. So, you might have heard these are being called acceptor molecules, charged molecules, or in this case, electron carriers. All right, they're all exactly the same thing. In our activity that we did in class, we saw them represented as little cars. All right. So the name of the electron carrier in photosynthesis. Remember, P for photosynthesis. So it's NADP. Okay. And in cellular respiration, we have 2, NAD and FAD. Okay? Explain why the electron carrier molecules are not included in the equation of cellular respiration or photosynthesis. So, you're probably wondering, that's a great question. So, what they do is their role is actually to carry energy or hydrogen ions, just like you've all told me in your photosynthesis sac, from one stage to another. The molecules themselves are not actually used within the reaction. So the hydrogen molecules are included in the equations because they're used, but the electron carriers are just transporting them from one stage to the other. They're not actually used to create or break down any of the products that are seen within those equations, and that's why. Okay, so I'll just jot it down here. So they carry the hydrogen ions, not directly required or used. Involved. Okay. Hopefully that clears that up. All right. Oh, can I do this in under an hour? I've got 7 minutes left. All right. So I have a circular diagram here with reaction A and reaction B. I'm not quite sure why they've got reaction C and reaction D. I think that reaction A and reaction C are the same, whereas reaction D and reaction B are the same. I don't know. If anyone can shed some light on that, that would be great. Please let me know. So we have ADP plus PI going into reaction C. A product is being formed and released, and we're creating ATP. Okay, so energy is being released. That's dreadful. I'm hoping you can read that, so I can't. So then if we have a look in reaction D and reaction B, it's taking in that energy, it's taking in the X molecule that was released from the first reaction, and then it's breaking down that energy into ADP plus PI. So energy is required. So if we go to the table below, so complete the following table by selecting the appropriate choices. Reaction A, so that's here. Is it anabolic or catabolic? We're going to come back to that column. I'll go to the last one. So is it endergonic or exergonic? So if you have a look here, it's being released. Energy is being released. So exergonic. Exerve refers to exiting. Okay. Whereas reaction B, it requires energy. Okay, so it needs to enter it, so it's endergonic. Okay. So now we're going to go back to the other column. So we've got anabolic and catabolic. If we are building something up, so remember that's anabolic because of estrogen and steroids. I go for estrogen, by the way, so I'm not putting them down. They didn't really do it. Anabolic. Building up, we require energy. Okay. Building up. So therefore, if we're releasing energy, we're breaking bonds, so we're breaking it down. So therefore, A is catabolic and B is anabolic. All right, so I've just had a breakthrough. Reaction A here, they're referring to that as cellular respiration. Reaction B, photosynthesis. Which makes perfect sense. Okay, so then the name molecule X. This is obviously water, so if you make sure that you go back and you have a look at the reactions, the equations, sorry, and you have a look at the outputs and the inputs for both and see how they interact with each other. All right, so that's obviously water. So let's write that down here. In the diagram above, which reactions are condensation reactions? So, condensation reactions are where water is created. So, cellular respiration, it creates the water, so we would say reaction C. Water is formed, so that's occurring at the electron transport chain. And then reaction B, photosynthesis, has an input, obviously, of the water, but then also has an output, so we'll say B. Okay, great. Hopefully that helps. Please make sure that you remember to tally up your answers and send them through via Edmodo. Great, thanks guys. Keep up the really good work.